All right, let's get started. Thank you for joining us today on today's exciting talk by Dr. Bruce Bean on the effects of Suzetrigine on human sensory neurons. Given the audience today, there is tremendous interest on both the compound and the target for pain, for which the promise of sodium channel inhibition for pain relief has been reignited by Vertex's successful and clinically approved development of Suzetrigine. So first slide, please. I want to thank Gary Watkins, Director of Marketing at AnaBios, for putting together this webinar series. As a quick intro for those who have not joined us in the past, AnaBios is a unique contract research organization and biotech based in San Diego. We recover human organs from ethically consented donors and use the tissues or cells to conduct physiological assays in which we can test preclinical compounds for drug discovery projects. We have essentially redefined first in human. And working with AnaBios is as close as you can get to a clinical trial without actually doing a clinical trial. For those developing therapeutics for pain, AnaBios has been evaluating pain therapeutics for over 15 years. Dorsal root ganglia, as well as trigeminal ganglia, are recovered from postmortem donors with both normal condition and pain or neuropathy pathology. AnaBios provides services for the entire drug development pipeline, from target identification and validation through the provision of tissue samples for transcriptomics or proteomic work, or validation of sensory neuron modulation by tool compounds, to lead identification through supporting medicinal chemistry or evaluation of gene therapy constructs, to lead optimization and candidate selection. Okay, next slide, please. We've developed new assays for clients. A new target of increasing interest is Nav1.8. This channel has been known for over 2 decades, since first identified in the labs of Suleiman Dibhaj and Steve Waxman. However, it's been a challenging target given the difficulties to express it as a recombinant ion channel. Recently, several CROs have developed patch clamp assays around recombinant Nav1.8, boosting industry drug screening efforts. AnaBios has developed voltage clamp assays to assess Nav1.8 in human sensory neurons. This slide shows Nav1.8 current in both DRG neurons and trigeminal neurons. The graph shows both the voltage dependence of activation and of steady state inactivation. AnaBios can work with clients developing methods to downregulate the current, to validate the efficacy of modulation, and to assess the effects in the context of the other sodium channels. Next slide, please. At AnaBios, we've been developing assays around triggering action potentials with physiological stimuli. This slide shows the triggering of action potential trains by repetitive additions of mildly low pH solution. As you can see at baseline, a brief puff of low pH of 6.5 depolarizes the membrane and triggers a train of action potentials. This can be done stably. Application of high concentration of selective Nav1.8 inhibitors, Latigo 33 and VX548, i.e. Suzetrigine, inhibit action potential firing in these trains. But interestingly, while these compounds reduce firing, rarely do they fully block firing, even at concentrations much higher than their respective IC50s and voltage clamp. Finally, the next slide, please. We have developed other models, including the triggering of action potential trains by inflammatory cocktails. This slide shows the high degree of activity triggered by a complex cocktail inflammatory mediators. These models allow clients to evaluate the therapeutics in human relevant in vitro models of nociception as well as pain. We would be happy to discuss further. Finally, technical points regarding the webinar. First, there are handouts, PDFs regarding AnaBios services that can be downloaded from the handout section on the webinar control panel, which is on your lower right. Regarding questions, we would like to hold addressing the questions until the end of the webinar. To submit questions, please use the chat box in the webinar control panel. You can type in the questions at any point, and I will read the questions at the end of the presentation. So with no further ado, it's my great pleasure to introduce today's speaker. To many in this audience, today's speaker needs no introduction. Nevertheless, it's a delight to introduce Dr. Bruce Bean. Bruce Bean is professor of neurobiology at Harvard Medical School. He graduated from Harvard College with a degree in biology and worked as a lab tech in the cardiology research lab at Boston Children's, and did his PhD research in biophysics at the University of Rochester, working with Peter Schrager on the pharmacology of sodium channels and crayfish giant axons. He did his postdoc work in cardiac electrophysiology, working with Dick Chen at Yale University, and has had previously had faculty positions at University of Iowa and at the Bollum Institute at Oregon Health Sciences. His lab focuses on the understanding of diversity of ion channels in different kinds of mammalian neurons, and attempting to use that knowledge to develop new treatments for pathophysiological conditions, including epilepsy and pain. Please take it away, Bruce.
Thanks. Well, thanks, Richard, and thanks very much to Richard and Gary for arranging and facilitating the webinar. And this is a collaboration between the scientists in my lab and the scientists at AnaBios, who have developed such nice protocols for obtaining and dissociating human DRG neurons and allowing us to do pharmacology on the species we are most interested in, especially in the context of pain. So what I'm going to talk about is a project that was done collaboratively, using the neurons from AnaBios, doing the experiments in my lab, and exploiting the amazing Nav1.8 inhibitor, Suzetrigine, which I will refer to mostly as VX548 during the webinar, because that's when we first started using it, that was the terminology we used, and that's what's in most of our figures. So as we all know, the understanding of how action potentials work and how neural excitability works really rests on the foundation of experiments done by Hodgkin and Huxley, showing that action potentials in squid giant axon are generated by two different kinds of voltage-dependent conductances. For those of us who were lucky enough to be alive and interested in this in the 1980s, there was a real revolution, both from molecular cloning and from the patch clamp experiments, allowing us to record from mammalian neurons, which previously was very difficult to do, especially in voltage clamp experiments. And those experiments through the 1980s and continuing to now have shown us that the ion channels in most mammalian neurons are far more complicated than the two kinds of ion channels in the squid giant axon. So we now know that a typical mammalian neuron has dozens of different kinds of ion channels that work together in a coordinated way to give the pattern of action potential firing that's typical of that neuron. And of course, as a field, we've done a lot of work in trying to find inhibitors and in some cases enhancers of these ion channels, which have been very useful in understanding how they all work together to control excitability. The most interesting thing, I think, is that there are very different patterns of expression of different ion channels in different kinds of neurons. And that gives us the opportunity to selectively inhibit or in some cases selectively enhance the activity of particular kinds of neurons and try to do that for pharmacological benefit. So one of the most interesting distinct kinds of patterns of ion channels that was found in these experiments were in primary sensory neurons and DRG neurons, where experiments in a number of labs, Steve Waxman's lab, I'm showing here data from Toshio Narahashi's lab, but this was an experiment done in a variety of labs showing that small DRG neurons in particular have two different kinds of sodium channels that originally were distinguished by their sensitivity to tetrodotoxin. Tetrodotoxin, of course, was really characterized by Toshio Narahashi. And in using it, it was found that although in most neurons, tetrodotoxin blocks all of the voltage-sensitive sodium channels, in small diameter DRG neurons, there's a component of sodium current that's resistant to sodium to tetrodotoxin. And in work done in John Wood's lab and Steve Waxman's lab and other labs, it was found that the TTX resistant current is primarily due to Nav1.8 channels. Although, as Richard showed in the earlier slides, there's also a contribution of Nav1.9 currents, which are also TTX resistant. But under most recording conditions of sort of normal resting potentials, the current is predominantly from Nav1.8 current. We can talk more about that at the end of the webinar if people are interested in. As we all looked at the roles of these different kinds of sodium channels in the excitability of small diameter DRG neurons, it became clear that these currents have different kinetics, somewhat different voltage dependence, particularly in the inactivation, and therefore contribute differently to the overall excitability of small diameter DRG neurons. One of the issues here was that although tetrodotoxin is very effective for inhibiting the tetrodotoxin sensitive currents, it was difficult to find a very selective and potent inhibitor of Nav1.8 channels. And as everybody probably coming to this webinar knows, there has been this extremely exciting development in the development over many decades of Suzetrigine, VX548, by the team of scientists at Vertex. Suzetrigine is a phenomenally potent and phenomenally selective small molecule inhibitor of Nav1.8 channels in human DRG neurons. And I think for those of us who have been around for a while doing small molecule experiments, small molecule inhibitor experiments with ion channels, you can really appreciate this accomplishment of medicinal chemistry in developing a molecule that has this phenomenal potency and phenomenal selectivity. This obviously is something that was based on a huge amount of work by obviously very many skilled scientists. And of course, the most exciting aspect of this is that this molecule, Suzetrigine, has been found to be clinically effective in inhibiting pain in human patients and was FDA approved, initially at least for treating acute pain with postoperative pain. And this is described in the paper cited here, the Jones et al. Paper in New England Journal of Medicine from 2023, and then with more detail in the paper by Austin et al. From this year. So this slide shows the ability of Suzetrigine to reduce pain, postoperative pain, with significant improvement over placebo. But as you can see from this plot, it doesn't completely eliminate pain. And so that raises the question, since nociceptors, since the presumed action mechanism of action of Suzetrigine is to inhibit excitability of nociceptors, and since nociceptors express other kinds of sodium channels in addition to Nav1.8 channels, an obvious question is to look in some more detail at the role of Nav1.8 channels relative to other sodium channels in regulating the excitability of human nociceptors. And so that's what we set out to do in this collaboration with AnaBios. This rests on the very impressive body of work at AnaBios to develop protocols for doing enzymatic dissociation of neurons from human DRG to obtain healthy dissociated neurons. These were shipped to us, often arriving at 5 in the morning, not infrequently before a holiday weekend, but the people in the lab were ready for them, eager to record from them. And so once they arrive in the lab, we take the dissociated neurons, plate them onto small cover slips, usually in 24 well slides, and then keep them, usually trying to have relatively few cells on each cover slip so that we have lots of cover slips to record from. We typically select small neurons to record from. We wanted to do our experiments at 37 degrees because, as I'll show you, the potency of Suzetrigine depends on temperature, and of course the kinetics of ion channels depend very strongly on temperature, as do firing patterns. And so what we do is we worked out techniques where we could basically scrape off the cells and lift the cells off the bottom of the cover slip and then raise them up so that we can have them in front of a series of temperature controlled flow pipes. These have a internal diameter of about 250 microns. That allows us to do rapid solution changes between a variety of solutions and to do the recordings at 37 degrees. And systematically, in addition to doing the experiments with Suzetrigine and other sodium channel blockers, at the end of the experiment, we apply capsaicin to test whether the cell expresses TRPV1 channels and is therefore a likely nociceptor. And in fact, most of the neurons that we chose for recording from at the time we did the statistics here, 141 out of 149 neurons were capsaicin sensitive. And so the vast majority of neurons that we record from express TRPV1 and also express Nav1.8 channels and are very likely correspond to C fiber nociceptors. So I want to go into some technical details about doing the experiments with Suzetrigine. When we initially started working with this, we synthesized it. The structure wasn't available at that time. We were very gratified to find that that we synthesized it from the patent literature, guessing at what the structure likely was, later confirmed. And initially, we were very gratified in finding that when we put the compound that we synthesized on and tested a dose response curve, this was done on a Q-patch compact automated patch clamp, Sophion automated patch clamp system, which, by the way, is incredibly efficient at doing dose response data, especially with low concentrations and low amounts of solution. We were very gratified to see very potent inhibition of human Nav1.8 channels in a cell line with a KD of about 4 nanomolar. As we went on and did more experiments and tried different conditions, we then started adding pluronic, a poloxamer that's often used, particularly in automated patch clamp experiments, to enhance the solubility and potency of especially lipophilic compounds. And we found this made a huge difference in the KD that we got for inhibition of the cloned Nav1.8 channels. And you can see it reduced the IC50, the KD, by almost 20-fold or so. In the experiments I'm going to show you, we did the experiments with manual patch clamp, of course, on the human DRG neurons. We did them at 37 degrees. So we were thought it was important to go look at the potency of Suzetrigine with manual patch clamp under the same conditions of doing the solution application. And at this point, we just routinely include pluronic in all of the pharmacology that we do. And so on the left, in the bottom here, is a dose response done with manual patch clamp at 21 degrees with pluronic, and you can see how exquisitely potent VX548 Suzetrigine is for blocking Nav1.8 channels when all the conditions are sort of optimized here. About 80 picomolar IC50. Interestingly, with the same conditions, when we do the experiments at 37 degrees, there's a substantial reduction in the potency of Suzetrigine, although obviously it still works very potently. And this is actually typical of many compounds that bind very tightly to well-defined binding sites, where the off rate in particular can be very temperature dependent and faster at higher temperature. So under the conditions that we're going to do the recordings with the human DRG neurons, the IC50 is about 0.4 nanomolar. So we chose initially in doing the experiments on the human DRG neurons to use a concentration of 10 nanomolar VX548, which is about 25 times the IC50 and should therefore inhibit almost all of the Nav1.8 current in the cells. And this slide is really the main takeaway from what we did. And this is what we saw in the vast majority of human DRG neurons. If you put on, again, pretty much a saturating concentration of Suzetrigine, you don't inhibit the action potential completely. There's typically very little effect on the initial rising phase of the action potential. There is a reduction in the peak of the action potential and a reduction in the width of the action potential. And you can see these effects very cleanly if you do a so-called phase plane plot. If you plot the dV/dt as a function of the voltage, you see here's the maximum upstroke of the action potential, which is only moderately reduced by VX548. The peak of the action potential is reduced substantially, and the falling phase of the action potential is actually increased here, and that corresponds to the narrowing of the action potential. So the basic finding here is that clearly Nav1.8 channels contribute to the action potential, but not very much to the rising phase. They really, it's as if they really begin to activate just before the peak of the action potential. The Nav1.8 current contributes substantially to the peak of the action potential, and a lot, and continues to flow during the falling phase of the action potential, so that when you inhibit it, the action potential repolarizes more quickly. The data below here shows there's, these are typical results from the DRG neurons that we recorded from. Again, small diameter, Nav1.8 expressing TRPV1 expressing. Typically, the upstroke of the action potential is only modestly affected. There are a few cases where it's affected more. The peak of the action potential decreases. The width of the action potential decreases. Looking in a little bit more detail, we looked at the effect of inhibiting Nav1.8 channels on the threshold of the action potential. And so this is done with a sort of classic procedure for defining threshold by injecting short current injections and then finding the voltage at which the response just begins to switch from being subthreshold to suprathreshold, and then doing the same thing after blocking Nav1.8 channels with VX548. And what you see is that there's a very modest effect on threshold, in this case changed by just 2 millivolts. And again, fitting with the idea that the Nav1.8 channels really contribute not very much to the initial rising phase of the action potential, And the reason is that they activate relatively slowly compared with other kinds of sodium channels like Nav1.7 channels. And so then in the collected data here, you see that there's typically a small increase in threshold, but usually only a few millivolts. The other, the obviously very important question is what happens with repetitive firing? So if you inhibit rather than with a short current injection, which is useful for studying threshold, if you look at the ability of the cell to fire repetitively with a maintained stimulus, what we find is that in most cases, this cell can continue to fire repetitively after blocking Nav1.8 channels, but typically at a reduced frequency, at least over a long current injection, and with decrease in the size of the action potential. But you certainly don't completely eliminate the ability to fire repetitively in most neurons. One of the things we noticed in this is that although the ability of the cells to fire repetitively in a maintained way during a 1-second current injection was reduced, and so typically here in control with maintained repetitive firing, it's converted to firing, in this case, just 5 action potentials. One of the things that we noticed that seemed very odd at first, but then we kept noticing it, was that the frequency of firing early in the current injection was often accelerated, where the second action potential, the third action potential actually occurred faster than in control. So that stimulated us to go do, again, kind of classic protocols to look at the refractory period of the neurons before and after inhibiting Nav1.8 channels. And so I'm going to go and sort of give you a up close recapitulation of one of these experiments. So we start off determining the refractory period by, in a classic way, injecting 2 short current injections, sub-threshold by, I think, roughly 25% or 50% compared with the threshold, super threshold by about 50% or so compared with the threshold, so that the current injections are well super threshold, and then systematically decreasing the length of time between them to find the refractory period. So here's how that worked in control. In this cell, you decrease the interpulse interval, and then at this point, the current stops firing. So that's the refractory period. It's really a relative refractory period for this size of current injection. Now, when we do in the same cell, the same thing after inhibiting Nav1.8 channels with VX548, again, in the first action potential, you can see the peak smaller, the width is reduced, but we still are able to fire not only the first action potential, but a second action potential. And then as we decrease the length of time between the spikes, what we find is that the refractory period is actually decreased. And so the cell will now fire 2 action potentials in response to a current stimulation in which the second one failed in control. And this reduction in the refractory period can be, quite remarkable, as in the cell here. So here's the superimposed cells here, and you can see this was actually fairly typical, that in most cases the refractory period was actually decreased after blocking Nav1.8 channels. So at first that seems kind of paradoxical. I mean, the action potential is from sodium channels. You block some sodium channels. How is it that the refractory period gets smaller? We haven't really analyzed the reason for this in detail. The obvious hypothesis that we have is that the reduction in the width of the action potential activates less calcium current. It activates, therefore, less calcium-activated potassium current. The decrease in width also will activate less voltage-dependent potassium current. And of course, after the action potential, and this is very striking in these human DRG neurons, there's a very striking after hyperpolarization in these cells that is probably due to a mixture of voltage-dependent K channels activated during the action potential and calcium-activated K channels that are activated during the action potential. And the reduction of these conductances, and it's probably a mixture of them, the reduction of these K conductances as a secondary effect of the narrowing of the action potential, then probably reduces the K current that gives the after hyperpolarization, and then allows you to fire a second action potential earlier than in control. And I think this is a great example of how difficult it can be to predict what's going to happen when the waveform of action potentials is changed, because the action potentials and the AHPs and so forth depend so critically on a great many different kinds of ion channels, all of which are responding to voltage. And so whenever the voltage waveform changes, the activation of every ion channels change, and you can have these effects that you might not have predicted a priori. And so this is, obviously very interesting, because it's a way in which with a particular pattern of stimulation, the excitability of the neurons actually increases after you block Nav1.8 channels. Okay, so one question is, does the maintained repetitive firing reflect reverse use dependence of VX548? And this goes back to experiments that were originally done actually with another Nav1.8 channels, Nav1.8 inhibitor, A887802, in which we found that there was very strong relief of inhibition by A887802 by depolarizations. And in looking at this response, when we first started doing experiments with VX548, we saw the same thing, and that's shown here. So the phenomenon here is that if you give depolarizations, and if those depolarizations are strong and long enough, and as I'll show you, that varies a great deal among different Nav1.8 inhibitors, you relieve the inhibition by, in this case, VX548. And you can then assay that with a test pulse here to see whether the inhibition has been relieved by a preceding depolarization. And very strikingly, so here in the left, you see in control, and then after, in this case, 1 nanomolar VX548 to produce only partial inhibition. And this is now done with cloned Nav1.8 channels, doing the experiments at 37 degrees. What you see is there's a very effective inhibition by the 1 nanomolar VX548 simply from -80 millivolts. But if you precede that test pulse with a depolarization to a depolarized voltage, you can actually remove almost all, or about, in this case, maybe 75% or 80% of the inhibition of VX548 is relieved by this preceding depolarization. That effect varies quite a lot, as I'll show you in a second, among different Nav1.8 inhibitors, both in the voltage dependence of the relief and also in the kinetics of the relief. In the case of VX548, the relief of inhibition requires quite large depolarization, so it has a midpoint of about +33 millivolts. And probably even more important, occurs quite slowly, so that if you give a depolarization to +40 millivolts, it occurs with a time constant, the relief occurs with a time constant of about 300 milliseconds. It's shorter for larger depolarizations, but these are now outside the physiological range during an action potential. And so when we look at the relief of VX548 inhibition during action potential waveforms delivered at 20 Hertz, 37 degrees, we see that in fact there is no relief of inhibition by an action potential waveform at a physiological frequency. And so we get no reverse use dependence with VX548, although we do see it, for example, with A887802. Just again, just as a very slight digression here, we've gotten very interested in the structure activity of the different Nav1.8 inhibitors for producing this relief and then re-inhibition, the voltage dependence relief and re-inhibition of inhibition by the Nav1.8 inhibitors. And you can see that these vary really strikingly among the different compounds. And so, for example, Latigo 33 is relieved much faster than VX548 is, which by itself actually would probably produce reverse use dependence with action potential waveforms, except that when you look at re-inhibition by Latigo compared with VX548 or with A887826, the inhibition by Latigo is re-inhibited very quickly in the time between action potentials. And so with a physiological firing frequency, the inhibition by Latigo 33 is probably removed, but then restored very quickly during action potentials at various frequencies. So we've gotten very interested in trying to understand a little bit more. There are very nice papers by John Gilchrist and Brian Moyer at Latigo and by Jeremiah Osteen and his collaborators at Vertex doing chimeras and mutations to identify the binding site, which seems to be pretty much shared between VX548 and Latigo 33. The binding site is in the voltage sensor of the channel, and showing that the likely mechanism of inhibition is to stabilize the voltage sensor in the closed state. And then the idea for the relief of inhibition would be that if you drive the voltage sensor open with the compound bound, the compound unbinds. And then if you restore the voltage, the compound binds again. That unbinding and binding occurs with very different kinetics depending on the structure of the compound. And we're now planning to do a very systematic comparison of all the available Nav1.8 inhibitors that we have or can synthesize or can guess at the structure from the patent literature and try to get some idea of what the structure activity for this effect is among the different inhibitors. And I'll just say here, we were planning to do this with automated patch clamp with instruments from Sophion and subsidizing the overall study with our NIH grant. The NIH grant has just been terminated along with all other NIH grants to Harvard University. And we are now very interested in the possibility of trying to get sponsored research from non-NIH funds. I think this comparison will benefit the entire field. I think everybody will probably be interested in the results of this. And if it occurs to you that you might want to help us in support doing this, please contact me. Okay, so back to the human DRG neurons. That was looking at the effect on the cloned channels. And so the conclusion is that with VX548, and also with Latigo 33, although the inhibition can be relieved by depolarization, in the case of VX548, that relief does not occur at the time course of the action potential. In the case of Latigo 33, it does, but the current is re-inhibited very quickly. And so in both cases, you do not get reverse use dependence with repetitive firing. So going back with VX548, we were curious, okay, so you can maintain repetitive firing in 10 nanomolar VX548. Is that because there's still some Nav1.8 current left, or is it simply due to the other sodium channels that the cells have in them? And so to address that, we increased the concentration of VX548 to 30 nanomolar, 100 nanomolar, and found that in some cases at least, although there was typically some extra effect in inhibiting repetitive firing of increasing the concentration, along giving the idea that even a few percent of the Nav1.8 current can be effective in producing the repetitive firing. You can still see it even with massive and virtually complete inhibition of Nav1.8 current. So clearly, the other kinds of sodium channels in the neurons are playing a huge role. So to try to quantify the relative roles of the different kinds of sodium channels in the neurons, we turned to the technique of action potential clamp, which is very useful for quantifying the contribution of different kinds of sodium channels to controlling the firing pattern. And so the experiment here is to record in the cell the firing, in this case, repetitive firing in response to a 1-second current injection, and then use those recorded action potentials as the waveform, going in, switching the amplifier into voltage clamp mode, then looking at the activation of sodium current during the action potentials, during the first action potential, the second, the last action potential in the train of firing during repetitive firing, and then recording the ionic currents evoked by those waveforms, and then using pharmacology to dissect the overall current into components. And so here's a typical example of an experiment done like that, where, starting off with VX548, in this case at 30 nanomolar, you can see that it inhibits sodium current during all of the action potentials. The Nav1.8 current, defined by the inhibition by VX548, is just as big during the second action potential, and in fact the last action potential during repetitive firing, as it is during the first action potential. It's actually a little bit bigger during the second action potential and the last action potential. And the reason for that is that the rising phase of the second action potential is a bit slower than during the first action potential. The reason for that is that the rising phase of the first action potential is dominated by Nav1.7 current. And so in this experiment, after defining the Nav1.8 current with VX548, we then added on top of VX548 30 nanomolar GSAF1 and Nav1.7 selective toxin. And so then that defines the Nav1.7 current. And you can see, and this is a fairly typical example, that the Nav1.7 current is substantially larger than the Nav1.8 current during the first action potential. But in the later action potentials, the Nav1.8 current is dominant. However, there's still some Nav1.7 current even during the later action potentials. And if you look at the collected data here, you see that, again, very typically, the Nav1.8 current is present in all of the action potentials, including the later action potentials. The Nav1.7 current tends to be dominant in the first action potential and is reduced in size during the later action potentials. However, if you blow up this, so one thing, the electrophysiologists who've done experiments like this in rat and mouse neurons will be struck by, these are enormous currents. These currents, 40 nanoamps, 50 nanoamps, these are huge. And so when, for example, with GSAF1 defining the Nav1.7 current, that's enormous during the first action potential. It's smaller during the later action potentials, but still can be several nanoamps, which is plenty to contribute to the action potentials even for those. One of the things we found that when we were doing this is that doing the combination of VX548 and GSAF1, often there was still a sizable sodium current left, particularly during the first action potential. And so then in a subset of cells, after applying VX548 and GSAF1, we then applied on top of those, with those still present, 1 micromolar TTX, and found that remaining current was TTX sensitive, predominantly TTX sensitive. And so here's a collection of this action potential clamp experiments done on a variety of neurons. And you can see that during the first action potential, in general, the Nav1.7 current, the blue, tends to be dominant. Also, significant, but often smaller contribution of Nav1.8 current. And in the experiments where we tried it, in some cases, a very large contribution of this third channel type, which we are in the process of trying to identify. In the later action potentials, Nav1.8 current dominates, but in some cases, there's a significant contribution of Nav1.7. These results are actually very different than similar experiments that we and others have done, over many years in both rat and mouse DRG neurons. So we've now repeated some of those experiments in the mouse neurons to compare the components of current in the mouse cells with the human cells. And here's a comparison of these. And so what you see, and this fits with many decades of work in many labs in mouse and rat neurons, particularly in the Waxman lab, in showing that in the mouse and rat cells, it's really Nav1.8 that dominates. This is now looking at the current during the first action potential, and you can see that in the mouse neurons, it's really Nav1.8 that is the dominant current. And in contrast, if you look at the human DRG neurons, during the first action potential, it's really Nav1.7 and this third component of current that tend to be dominant. So to summarize these experiments, I think key findings here are that the human capsaicin-sensitive DRG neurons have very large sodium currents from both Nav1.7 and Nav1.8 channels. The inhibition of Nav1.8 channels alone, which can be done so beautifully by Suzetrigine, reduces excitability but does not abolish it. The inhibition of Nav1.8 can have some unexpected effects, including the paradoxical increase in the firing frequency due to the shortening of the refractory period. And I think very importantly, in trying to understand how we use data in rodents to predict effects on human DRG neurons, there's a big difference in the contribution of Nav1.7 and Nav1.8 currents in the two species, and human DRG neurons have a much larger contribution of Nav1.7 channels to overall excitability than we see in mouse neurons. And really, kind of interpreting, speculating here, it's very tempting to make a correlation between the partial but incomplete inhibition of nociceptor inhibition, nociceptor excitability inhibition by VX548 with the partial but very incomplete inhibition of overall pain sensation in the patients with postoperative pain. And because VX548 is so potent, this may indicate that there's going to be a ceiling of inhibition of nociceptor excitability by even the most potent Nav1.8 inhibitors, if those are used by themselves. So one obvious thought would be, because of the strong expression of Nav1.7 channels, is to combine some Nav1.7 inhibition with Nav1.8 inhibition, which should be much more effective in inhibiting the nociceptor excitability. Another avenue that we are exploring, particularly in experiments collaboratively with Clifford Wolf's lab, is to combine Nav1.8 inhibition with enhancement of Kv7 potassium channels, which are expressed, we find are quite heavily, strongly expressed in some human DRG neurons. And so if you, for example, in this human DRG neuron, if you enhance Kv7 current with the xenon compound, xenon Kv7 enhancer Zen1101, you, by enhancing the Kv7 current, you can greatly change the rheobase for firing action potentials. And if you combine that with inhibition of Nav1.8 with VX548, you can reduce the firing to a single action potential over a very wide range of current injection. And we are exploring this in more detail in collaboration with Clifford Wolf's lab and with some medicinal chemists, whose names I'll show in a second, to try to find single compounds that would combine, that do combine Nav1.8 inhibition with Kv7 activation. So this work, the main work that I showed you was this very productive collaboration with the scientists at AnaBios, people in my lab, Akie Fujita, Suyeon Jo, Patrick Valley, who's now joined the scientists at Sophion, Tommaso Sorno and Robin Stewart were the people who did the main recordings from the human DRG neurons. The team of scientists at AnaBios who have developed the techniques for isolating these human DRG neurons that are really remarkably robust in lasting, giving not only very nice recordings, but also lasting for many days. I didn't talk much about it, but we are now trying to make use of some of this information to develop new compounds that would combine Nav1.8 inhibition with Kv7 activation or with Nav1.7 inhibition. And we're doing that with some collaborating medicinal chemists. We are taking advantage of automated patch clamp to characterize these compounds, and then working very closely with Clifford Wolf's lab, and then trying to going to in vivo testing of these. This work up until 2 months ago was funded by the NIH. That funding has been terminated with extreme prejudice. And we are, especially as a result of that termination of NIH funding, particularly grateful to our collaborators, AnaBios, for furnishing us with cells, and also with our collaborators at Sophion Bioscience, who are very stepping forward to enable us to continue to use the automated patch clamp for some of the development of novel compounds. So thanks very much for listening, and I think we now have some time for questions.
Thank you, Bruce, for that presentation of amazing work. And yes, we have many questions. So I'm going to start. The first question actually is from Dan Beecham, and he remarks that it's great to hear your voice again. But he does ask, I guess in context of your lifting the neurons off the cover slip, if whether or not you forego the use of like laminin or other ECM coatings, and have you ever done any comparison studies with ECM present?
Sorry, with what?
With ECM or adhesion coatings.
Oh, I see.
Have you done that in comparison?
Yes. Unfortunately, I'm the wrong person to ask. I will say this, that we, Akie, Suyeon, Tomas, and Robin spent a lot of time experimenting with different solutions and cover slips, ways of attaching them to the cover slip and so forth. do we use laminin? Unfortunately, Robin's down at the other end of the lab, or I would shout out to him. So I think the methods in our PNAS paper that appeared recently will have that. And please email me if you're interested in that, and I can get the details on that from the people in the lab. My apologies for not remembering that. It's one of these things as a PI that you see everything's working great and you don't ask too many questions.
Thank you. So the next question, actually, I'm going to ask two, is from Mike Gold. First of all, have you tried recording from DRG neurons and current clamp at more depolarized potentials? I guess there was some slide that was at -75, and in the presence of TTX. And Mike's second question, let me just jump to it. More directly, as implied by the current clamp data presented, human neurons contain TTX sensitive currents. Have you ever tested the potency of AX548 on these other currents, the TTX sensitive currents?
Okay, yeah, so interesting questions. With regard to the resting potentials, we did the recordings we did the voltage clamp recordings. So for example, with the action potential clamp recordings, using the cell zone firing. And so that was done from the natural resting potential of the cells, which varied, but was typically, between -60 and -80 millivolts or so. The VX548, let me just think a second. It is, as in the data from Vertex, extremely selective when tested on cloned channels. Now, did we ever test it? I would say everything in the human DRG neurons fits with perfect selectivity over the TTX sensitive current in the cells. But it's a little bit, hard to, and I guess maybe the most relevant way of looking at that is in the AP clamp experiments, although we've done it with steps also, where if you look at the AP clamp experiments and you look at the time course of the currents, you see a very, striking difference in the voltage dependence and the kinetics. The VX548 sensitive current activates slowly, inactivates slowly, and has the voltage dependence expected of Nav1.8 current with no obvious contamination from rapid TTX sensitive sodium current. So I would say everything in the human DRG neurons fits with no inhibition of Nav1.7 or other TTX sensitive currents. Did that answer the question? Richard, you can tell me if there was another part to that.
I did not see another part to it, and I believe that answers the question.
Okay,
so
Mike and anybody else,
Mike,
feel free to email me, and we can discuss this more.
So I just want to comment, and we are at the top of the hour, but Bruce has graciously said that he can go beyond the hour of the talk, and so further questions can be asked. And so for those who have also the time to continue attending and add the questions, we will go further. So the next question is from Andrea Surison, and he asks, has any of the studies recorded the same parameters in the IPS-derived sensory neurons to evaluate, how these translate from the primary to the IPS and vice versa backwards?
Yeah, so great question. We have worked over the last, I don't know, I guess 5 years or so now, maybe more, in collaboration with Clifford Wolf's lab and with Ilyas Sinjic, who was at NCATS and now is at Fujifilm. And Ilyas and Clifford's lab worked together to improve ways of differentiating human iPSC cells into having RNA expression matching that of nociceptors. And they published that. In my lab, we contributed to some of the electrophysiology of those neurons. And one of the surprises was that electrophysiologically, we saw in most neurons no Nav1.8 current. Every once in a while, there'd be an iPSC-derived sensory neuron that had some TTX resistant current that could have corresponded to Nav1.8, but it was very rare. And so, so because of that, I think at this point at least, the iPSC-derived nociceptor-like neurons have an issue that they have very little Nav1.8 current. And so, so it's not really possible to do the comparison.
Okay, thank you. So the next question, and of course, you talked about this. This question gets at what you've been talking about for the last few slides, but I'll ask. It's from Adi Rava, and do you think that the compensatory upregulation or functional redundancy among the other NAV subtypes could explain the limited efficacy in the phase 2 trial? So specifically referring to the contributions of the various channels and their potential to compensate for each other. And of course, you've been addressing this. I don't know if you have some further comments to this question, which is a very important
Not
one.
really. I mean, no. And I guess what I would say, I think it's a very interesting question in a number of ways. First of all, one thing I would say is that we don't see any indication in our experiments that there are large, at least, changes over days in the relative contribution of currents in the neurons. Now, we can't rule it out. We absolutely can't rule it out. And of course, both in these experiments and in experiments that everyone else, we and everybody else does with mouse and rat neurons, once you sever the nerve and you take the neurons out, things are going to start to change. What I can say is that we see very little difference. So we get the neurons from AnaBios cold, basically. And if we start recording from them right away, we see very much the same pattern of combinations of channels as we see, over the next few days. So there's no obvious time-dependent changes. Now, that clearly does not mean that there could not have been changes, from the time that the dissection was done. So that's one thing. The other thing is, and I think this is something that all of us are going to be interested in, with long-term use of any sodium channel inhibitor, there well could be some sort of compensatory changes in excitability of the neurons. And of course, there's a vast literature on sort of homeostatic mechanisms that you see in neurons, and it's very dramatic, for example, in neurons. If you completely inhibit firing with TTX in a cortical neuron, then you see all sorts of changes in intrinsic excitability, synaptic currents, and so forth. And so I think it's absolutely something that, especially with long-term use of any very potent sodium channel inhibitor, that will be, just important to pay attention to, because there could be compensatory changes that you might not see, if you're applying it post-operatively just for a day or two.
Thank you. And from the point of view of if someone were to ask, like the differences since we did ship them to you cold, et cetera, and you're doing it in your lab, I would say that your results are in broad strokes very similar to what we observed too. So we don't think that there's a, like an effect by, having gone to your lab. I will, so there are more questions. So here's a question from Suyon Sim, which is very different concentrations were used in the re-emission study. 1 nanomolar VX548 versus 300 nanomolar Latigo 33. So, essentially, what's the importance, I guess, of the difference in concentration, if that had any impact?
Yeah, that's a great question. I mean, I think I'll refer you. That gets a little bit complicated. The intrinsic IC50 is quite different for Latigo 33 than VX548. And we had a paper in Journal of General Physiology, which I think is now just available online, in which we did a detailed comparison of some of those, including looking at different concentrations of the compounds. So the reinhibition, that's true. And the reinhibition with Latigo is at the higher. It was done at a higher concentration because the IC50 is quite different. But
right.
Basically what we see is that the relief of inhibition does not depend on the concentration of compound. The reinhibition is linearly related to the concentration of compound. And we interpret that to mean that the relief doesn't depend on the concentration, because that's just sort of intrinsic to the interaction of the channel with the compound, and that the compound probably comes completely off the channel, and then with the reinhibition, it all depends on the on rate, which is very strongly concentration dependent.
So here's another question from Dan Beecham. So yeah, this is an interesting question. So where do you think that the Nav1.8 are located spatially in the small diameter neuron? Do we have any hypothesis about the 1.8 function in vivo in terms of encoding the synaptic output to the dorsal horn?
Right, great question. I have nothing to say about that, in addition to what has been done in the literature, primarily in rodents. I would, as far as I know from the literature and from talking with people, we really don't know what the role of Nav1.8 is at the nerve terminals and on the action potential dependent release of glutamate onto dorsal horn neurons. And I think that's a really interesting question because, as I showed, the action potential gets shorter and narrower with VX548. And so if that occurs at the nerve terminals, at the boutons where the glutamate is released, then that can be one mechanism by which VX548 is going to reduce synaptic transmission in addition to reducing excitability of the cells. But as far as I know, we basically have no information on the relative contribution of Nav1.7, Nav1.8, or the other sodium channels at that point in the neuron.
Right, thank you very much. So just a note from Michael, and I think he's representing the thoughts of both the academic as well as industry researchers, which is about the grants. So, of course, for the field and for all scientists, we're lucky if we lucky we make it through the current administration. I'm going back to a scientific question. Does the Suzetrigine enhance the closed state?
Yeah, I think yes. I mean, that's I think the way to look at it. From the state dependence that we see, and interpreted by the experiments that John Gilchrist and Brian Moyer and Jeremiah Osteen did. The mechanism of action, really, the VX548 is a gating modifier. It works by binding tightly to the closed state of the channel and stabilizing the closed state of the channel. And if you activate the channel with a strong enough depolarization, then you destabilize the binding. So in a lot of ways, it's sort of exactly an inverse of classic state-dependent stabilization of inactivation by drugs like lidocaine or carbamazepine. Bind tightly to the inactivated state, stabilize the inactivated state. Here it binds tightly to the closed state and stabilizes the closed state.
Actually, just a follow-up question to question is, as TTX inactivates 1.8 at 30 nanomolar, I'm sorry, millimolar, can Suzetrigine inactivate other voltage-gated sodium channel subtypes in the high concentrations?
The answer to that is yes. And again, I wish I had Su-Yun and Nakie and Robin and Tomas sitting next to me, because they will remember the details better. But in essence, we have gone to very high concentrations of Suzetrigine in looking at Nav1.8 in mouse neurons, because the potency in the mouse Nav1.8 is greatly reduced compared with the human. It still inhibits, and so we can go to micromolar concentrations and get inhibition of Nav1.8. But in doing that, once you get up to micromolar concentrations, you see, for example, inhibition of some potassium channels also. And so absolutely, there's a point at which the selectivity is imperfect.
So regarding the other subtypes, did you detect, this is another question, so did you detect any TTX resistant but non 1.8 current in your recording conditions?
In our recording conditions, not usually. Mm-hmm. But we didn't look at it that way. And so let me say two things about that. One is that as Richard, as you showed, and we certainly see the same thing, there's now 1.9 current in the neurons, no question about it.
Exactly.
To see it, as all of us know who have studied it in mouse and rat neurons, you typically need to hold the membrane at strongly negative voltages, negative to like -80. So in our experiments, we did almost always things at the natural resting potential of the cells. And and at those voltages, we typically saw very little obvious Nav1.9 current. At the end of an experiment, if we applied VX548, plus GSAF1 or plus TTX. However, particularly early in an experiment, we've done this more recently, if at the beginning we hold the membrane a bit more negative, we can get huge Nav1.9 currents. So they are there, as in the experiments that Richard showed. And as in the human neurons, as in the mouse and rat neurons, there's a puzzle. Where are they at normal membrane potentials? Under what circumstances do you see them at normal membrane potentials? And as people know, there's literature showing you can upregulate them with inflammatory conditions and so on and so forth. But it's still a little bit unclear. I think how much they and under what conditions they contribute to the excitability, at least of the cell bodies.
Right, exactly right. Thank you. So another question regarding your observation of the shortening of the refractory period. So do you have any thoughts on how this might actually translate in terms of its efficacy in vivo?
Short answer, no. I think it's a really interesting question, and it gets into the question of in vivo, exactly how the patterns of action potentials are generated by the peripheral, by the peripheral terminals in response to the stimuli that lead to the pain sensation, and that gets kind of complicated. We don't have I think a very good picture of exactly what's going on in terms of the waveforms as action potentials are being generated. So I think it's a very interesting question, but don't have any clear data or insight into that.
Mm-hmm. Thank you. Here's an interesting question: when you observed the difference between human and rodent on regard to 1.7. So, as David Akos observes, the dominance of 1.8 in rodent is due to depolarized resting membrane potential in the cell bodies. So, or that is in culture. In vivo, 1.7 actually dominates for action potential generation in mice. I believe he's, of course, referring to their work on the knockout. Could this part of this be part of the explanation of the species difference that you're discussing and observing?
Yeah, it could be. Yeah, I mean, right. So the one thing I would say, I would disagree slightly with the statement that the dominance of 1.8 in the cell body is because of depolarized resting potentials, because I think that even with holding the cell body at strongly negative voltages, that there's still still a dominance of Nav1.8 in the action potential. Now, obviously, things are different in the propagation of action potentials in the axon and looking at the neuron as a whole. And it may be somewhat condition sensitive and so forth, whether you're looking at acutely dissociated, after time and culture and so forth.
Now, so here's an interesting question regarding the kinetics and the issues of kinetics and the potential. Well, when screening drugs with optical methods, is this result in the discovery and rediscovery in closed and steady state inactivators? Do you have any thoughts of the methodologies that might, at scale permit screening for use dependent and open state blockers?
Yeah, great question. And, short answer is yes. I would promote screening with automated patch clamp, because what's great about that is that you can do those kinds of experiments in as much detail as you like. And in fact, in our screening, using the Cube automated patch clamp, the 384 channel, we use for that reason, we use a variety of protocols, including protocols designed to quantify the voltage dependent relief, to quantify use dependent using AP like stimuli and so forth.
Great, thank you. So here's an interesting question as well, whether or not if Suzetrigine, given it's maybe more effective because of what you're observing in terms of the 1.8, would you think it might be actually more effective in veterinary medicine?
Let's see, good question. I guess I don't have the expertise in knowing. I mean, if I remember correctly from the Vertex data, I think the dog channels are particularly insensitive to VX548. So I'm not sure, at least there, that particular compound is going to be, effective. Right. I don't have enough knowledge of what's known in, at least the normal, animals to say anything more than that.
So several more questions with regard to the refractory observation or the interval for action. I'm going to phrase it in this one. So if 548 decreases neuronal excitability, but paradoxically increases firing frequency, what's more important in modulating perception of pain? Total number of spikes or inter-spike interval?
I think that's a very interesting question, and again, I would say sort of above my pay grade. in terms of knowledge and intuition. You know, probably the frequency of action potential generation is very important. But again, exactly how that is generated by the painful stimulus, you know, probably varies a lot depending on, you know, is it painful cold? Is it, you know, something like capsaicin? Is it tissue damage? Is it, you know, protons released from tissue damage? You know, those are all going to vary because they're all going to produce a depolarization that's then going to produce some, you know, generate some frequency of firing of action potentials. And that probably is going to vary a lot depending on what the stimulus is. Obviously, a particularly interesting one is spontaneous firing that, you know, you can occur in nociceptors in conditions of neuropathic pain, especially following, you know, following tissue damage, you know, long term. And exactly the pattern of firing and so forth there is probably going to have a big effect. I would say in general, I mean, my general feeling would be that the reduction in the refractory period may be more of a sort of electrophysiological curiosity, because I'm not sure the refractory period, you know, comes into play in the generation of the firing of the neurons as much as it would like in a central neuron or something like that was firing very rapidly. Because in general, C fiber nociceptors fire relatively slowly. You know, 20 hertz would be a fast firing frequency.
Okay, thank you. So, you know, we've gone beyond like 25 minutes beyond the hour. So I really appreciate the generosity in answering all these questions, which is, and you know, the audience is obviously very engaged and still asking. I'm going to ask just a couple of more. One would be to comment on the differences between a normal DRG relative to DRG from potential donors that have pain.
We don't have enough data yet. And I think it's a really interesting question. But I can't say anything from our own data. I think probably collectively, you know, Richard and the other scientists at AnaBios have more experience with this. There's some nice literature from Rob Giroux, from Ted Price, you know, looking at differences in human DRGs, you know, taken from DRGs that, you know, innervated people who had, you know, diabetic neuropathic pain and other conditions. So there's some information on that in literature. We do not have enough recordings to say anything about that.
Okay, thank you. One quick question follow-up on that is the idea, your observation about the KV7 combo with the 1.8. Have you are you planning to, or have you looked at any kind of disease models and look at spontaneous activity?
We have not looked at we have not looked at spontaneous activity. We have not looked at disease models in humans. In collaboration with Clifford Wolf, as I mentioned, we are working in this direction in a variety of mouse models of pain. But I would leave that for another day because that's really data from Clifford's lab to go into that in detail.
Yes, thank you. Yeah, as I said, we're getting really, so I apologize if I haven't gotten to your question. I'm going to ask this another question about combination, and then I think we will have to end it. But again, I appreciate all the questions and the interest. So there is a question about the combination. Well, specifically, if you use super maximal dosing of Suzetrigine, that might be limited by safety concerns. So would you think that combination therapy, you know, whether or not it's the 1.7 or the KV7, achieve better analgesia in patients with chronic pain?
Short answer is I think so. And I think, I think this is something that as a field is very well worth pursuing. I would say based on what we see of the contribution of all the various channels to the overall excitability of the human DRG neurons, that it's likely that combining inhibition of 1.7 with 1.8, that likely that you could have less complete inhibition of each one and still have very effective inhibition of excitability. Similarly with combining Nav1.8 inhibition with activation of Kv7. I think it's likely. I think we all need to do the experiments. And ultimately, of course, it's going to depend on the clinical tests.
Here's one. I'll ask this one last question because I haven't been able to ask the question from this audience member. But the question is whether or not how long the protocol lasts and how long the VX548 affects exponential generation. I'm not quite certain if this means really long-term application or referring to the reverse use dependence. But I absolutely.
Yeah, I mean, I guess if the question is about VX548 reversibility, it does reverse. It's it got a very high affinity, but it can be reversed, especially at 37 degrees. And that's actually one advantage of doing experiments at 37 degrees is that the reversibility of things like TTX and VX548 is much faster. So it's easier to do experiments if you want to do different combinations. So it behaves, the VX548 behaves in a very rational way in that it comes on, comes off, so it's certainly reversible.
Okay, well, thank you so much, Bruce, for a fascinating both presentation as well as all the addressing all the questions. It obviously indicates how important the topic is really to develop a drug. Again, I want to thank both you. I want to thank everybody who's attended the webinar. It's much appreciated. We will be doing more pain webinars later this year, we hope. And so again, thank you very much, and please attend any future webinars we have. Thank you.
Thanks. And I just say to everybody, if I didn't get to your question or you have more questions, feel free to email me. I mean, I love this stuff, and it's great to hear questions always stimulate new experiments. So this has been very stimulating.
Thank you, Bruce. Fantastic.
So long.
Bye-bye.